Okay, good afternoon. You're very welcome to this seminar as part of the School of Agriculture and Food Science seminar series. My name is Lorraine Brennan and I'm going to chair today's session. This seminar series is a series of talks that cover a range of topics from agriculture to human health, and we have a range of local and international speakers. And today it gives me great pleasure to introduce a local speaker, Dr. Martina Wallace. Martina will speak for about 35 minutes and then we'll have time for questions. So I encourage you to put your questions into the Q&A box at the bottom of the webinar. So Martina joined the UCD as an ad astra fellow and assistant professor in human nutrition in May 2020. She originally carried out her BSc here in UCD and also performed her PhD here in UCD. And in 2013, she went to the US to complete her postdoctoral research in the University of California, San Diego's bioengineering department. And there she focused on the role of amino acid and lipid metabolism in obesity. So today Martina is going to talk to us about tracing insights into branched chain amino acid utilization in adipose tissue. And I'm very much looking forward to hearing about this. So over to you, Martina.
Thanks, Lorraine. I'll just pull up my slides now. Okay, so thanks very much for the intro. So as Lorraine mentioned, today I'm going to talk about the work I did while I was in San Diego, where I looked at branched amino acid utilization in adipose tissue. So Lorraine somewhat covered this, but I just wanted to start by giving you a brief overview of my background and how I came to here. So I did my degree here in UCD in biochemistry. And that's where I became quite interested in metabolism. And then I went and I actually did my PhD with Lorraine's group here in UCD, where I learned how to apply metabolomics for biomarker discovery and also for looking at cell function, specifically in relation to diabetes and obesity. And then in 2013, I went and worked in Christian Metallo's lab in UCSD. And so his lab specializes in applying stable isotope tracing to look at metabolism and metabolic flux. So here I use these technologies to look at adipose tissue metabolism in obesity, among a number of other diseases such as cancer and also macular degeneration. And then last May I came back here, and now I'm setting up my lab here to continue some of the work I did in San Diego on branched amino acid metabolism, and look further at the interaction between diet and metabolism. So we all know that metabolism is an essential function to support growth and energy generation in all living cells, and that it plays a driving role in many diseases. And really, by uncovering in-depth knowledge of metabolic pathway architecture and also flux, it can be used then to promote optimal health in humans and animals. And also classically, this kind of knowledge has been used to drive synthesis of valuable metabolites and macromolecules in things like bacteria and yeast. So these are really the main kind of questions that drove development of methodology in this area. And so when you're thinking about studying metabolism, there's a number of different ways that it's classically approached. So the more classic way of looking at it is looking at isolated enzyme kinetics. And from this, you get a really good idea of detailed knowledge of one enzyme and how specific mutations can change that enzyme's reaction, but it lacks the benefit of studying how reactions perform in a network. So because of this, now it's much more common to use things such as metabolomics or measuring metabolite levels. And this is really useful because you can get a really broad fingerprint of how metabolites are changing in a specific biofluid or cell under different conditions. And it's very good for hypothesis generation, but it does have some limitations. So for example, if you have an increase in metabolite C, you don't know if that's because of a decrease in the activity of enzyme Y or an increase in the activity of enzyme X. So it lacks that information about direction or identifying the specific node of metabolism that might be perturbed. And in addition to this, abundance of metabolites doesn't always align very well with flux. So you can have a pathway with quite high abundant metabolites, but that doesn't mean that the greatest flux is going through that pathway. So to fill that gap, this is where the use of stable isotope tracers and metabolic flux analysis comes in. So this allows you to get more information on directionality of metabolism, new pathway architecture. So by following where these heavy labels go, you can find out if novel products are being produced, and also relative substrate utilization, and you can calculate quantitative flux through pathways. And so I spend most of my research, I suppose, kind of in between these two areas of applying isotopes in cell culture in animals, and then using more classic metabolomics for human studies and also animals. So today I'm going to talk about branched amino acid metabolism. So branched amino acids are well known as essential amino acids in the diet for protein synthesis. And they also kind of augment that role by signaling via mTOR to promote growth. And they're also, as they provide an important nitrogen source in cells for synthesis of other non-essential amino acids. But in addition to their importance in protein synthesis and nitrogen provision, they're also, what's maybe more underappreciated is that they can also act as an important carbon source for cells. So after the nitrogen is taken off, these branched-chain keto acids can be metabolized within mitochondria to generate acetyl-CoA and succinyl-CoA for energy generation in the TCA cycle. And disruptions in branched amino acid metabolism have been associated with a number of different diseases. So it's well documented now that levels of these are elevated in patients with cardiovascular disease or obesity and insulin resistance. Alterations in the pathway have also been associated with cancer formation and progression. And there's also at least 16 different inborn errors of metabolism or diseases associated with mutations in these enzymes that can lead to varying degrees of physiological dysfunction. So understanding this pathway in depth is important for a number of different reasons. But when I first came to, or started as my postdoc, I didn't go there aiming to study branched amino acids, but just more to understand carbon metabolism within the adipocytes or adipose tissue. And the reason for this is that adipose tissue is a really important regulator of systemic metabolism. So as well as its more well-known function as a storage of excess carbohydrates and dietary fatty acids, it also plays an important endocrine role through the release of hormones such as leptin. And what's emerging more now is that different adipose depots can release signaling metabolites that impact the function of tissues around them. And of course, there are many different types of fat, ranging from the more storage-orientated white adipose tissue to the more mitochondrially active, heat-generating brown adipose tissue. So in order to study adipocytes then, we decided to use the 3-3L1 adipocyte culture system. So basically with these cells, you grow them as pre-adipocytes first, which are kind of like proliferating fibroblasts. And then by adding an induction cocktail, you can promote differentiation to these lipid-laden, more mature adipocytes. And what we did is we took these adipocytes then and we changed the some of the nutrients in the media to stable isotope versions. So for example, glucose, glutamine, and branched amino acids. And then we looked at the incorporation of these isotopes into downstream metabolic intermediates using GCMS, where you can see if with the mass increment in that specific metabolite, how much isotope has been incorporated into it. And from there, we calculated relative substrate utilization. And so this is an example of the kind of data you would get from this experiment. So basically, if you trace a cell with 13C labeled glucose here, where all the black dots are 13C carbon, well then this will get broken down into a fully labeled acetyl-CoA, which can then be used to make citrate. So when you look at the mass isotopomer distribution of citrate then after tracing with glucose, what you typically see is this really distinct mass increment of 2 coming from incorporation of that acetyl-CoA. And that enrichment of citrate can tell you about how much glucose it's using versus other carbon sources. And then in addition to that, you can also follow this carbon all the way to fatty acids and de novo synthesized fatty acids. And what you get during fatty acid synthesis is this sequential kind of addition of acetyl-CoA units. And then in the pattern of the mass isotoper distribution pattern, you see these mass increments of 2 coming from that fully labeled acetyl-CoA. So it has a very distinctive pattern. And you can use that data then and apply isotoper spectral analysis, and then you can quantify the contribution of different substrates to acetyl-CoA pools within the cell, and also calculate fatty acid synthesis rates. So after doing this in the 3-3L1s and looking at how metabolites or how metabolism changes from the pre-adipocyte to the differentiated adipocyte, what we found was that although in most proliferating cell types in culture, so a lot of different cancer cell lines, and also in pre-adipocytes, the majority of carbon used for de novo fatty acid synthesis comes from glucose here in the light blue and glutamine in the dark blue. But when it comes to differentiated adipocytes, there's a large amount of that carbon that's unaccounted for. So they're making these fatty acids from some other source. And of course, we thought of branching amino acids at the time because there was a number of papers showing that gene expression of this pathway is upregulated with differentiation. So when we traced the cells then with isotope labeled versions of leucine and isoleucine, what we found is that they can account for as much as 30% of the acetyl-CoA used to make fatty acids in these cells. So they really use a lot of branching amino acids as a carbon source. So this kind of indicated to us then that adipose tissue may be a really important site for branching amino acid usage. And so when we were doing this originally, it was very well established that branched chain amino acids are elevated in obesity and diabetes, but the role of adipose tissue in driving that was less well established. However, there was some evidence showing that that activity in adipose tissue in genetic mouse models could alter circulating branched amino acid metabolism, and also that gene expression of this pathway was decreased in adipose tissue from obese people. Well, one thing that wasn't known is that no distinct factors that played a role in adipose tissue dysfunction in obesity had been shown to regulate branched amino acid catabolism. And so we came across this paper that showed that combined treatment of adipocytes with the pro-inflammatory cytokine TNF-alpha and hypoxia was the best in vitro conditions to mimic what was seen transcriptionally in insulin-resistant adipose tissue in vivo. So that this was a really good model for recapitulating some of the in vivo phenotypes. So we decided to apply this to our cell system. So to do this then, I took the adipocytes and I exposed them to hypoxia or 1% oxygen for 48 hours and added tracer after 24 hours. Or I exposed them to TNF-alpha for 48 hours and again added tracer halfway through. And what I found was that when I looked at citrate enrichment from branched amino acids as an indicator of how active the catabolic pathway was, I found that TNF-alpha had caused a subtle decrease in catabolism, but that hypoxia really potently suppressed the use of branched amino acids for the TCA cycle. So we then wanted to see how long does it take the cells to recover after this. So oftentimes in vivo, of course, tissues can be exposed to varying oxygen levels, and we wanted to see how quickly the different main carbon sources such as glucose, glutamine and branched amino acids recover after going through a hypoxic insult. So what we did is we put some of these cells back in normal oxygen and added a tracer at the same time to see how metabolism changed. And so when we looked at citrate enrichment from glucose, you can see that actually this was minimally changed with both hypoxia and reoxygenation. So although total flux of the TCA cycle was decreased, the relative contribution of glucose to that citrate pool was maintained with hypoxia. Whereas the contribution of glutamine increased with hypoxia, which is quite well established in other cell lines, but that it recovered relatively quickly and went back down to normal levels. However, when we went and looked at branched-chain amino acids, we can see again that it's significantly suppressed with hypoxia, and it doesn't really fully recover in those 24 hours after hypoxia. So this indicated to us that this pathway seemed to be more susceptible and more sensitive to changes in oxygen than some of the other pathways we were looking at. And then in addition to that, when we looked at how these different carbon sources then contributed to new fatty acid synthesis, you could see that after hypoxia, when they're put back in normal oxygen, the contribution of glucose actually increases. The glutamine recovers and goes back down to lower levels very quickly, but again, the branched amino acid contribution to fatty acids does not recover. And then finally, we wanted to identify why or what the main node in the branched amino acid pathway was that was causing this. So we looked at BCKDH, which is the rate-limiting enzyme in the breakdown of branched-chain amino acids, and we found that levels of this enzyme were decreased in hypoxia and that they failed to recover with reoxygenation. Okay, so overall then this data showed that the pathway does seem to be quite sensitive to low oxygen levels, but we wanted to see what happens in vivo and if branched amino acid contribution to the TCA cycle really was decreased in obese adipose tissue, which is thought to be more hypoxic. So to do this then, we took black 6 mice and we fed them either a low-fat diet or a high-fat diet for 12 weeks, which is a classic model of obesity in mice. And then for the last 3 weeks, we replaced 25% of the leucine and valine in their food with 13C labeled versions of this. And then we tracked where this went. So what we found is that when we looked into citrate enrichment across all the different tissues, that this was decreased by a high-fat diet. So this indicates that there was pretty much a whole body suppression of branched-chain amino acid catabolism in response to a high-fat diet, and also showed us that actually these are the enrichment levels across such a range of tissues showed that this is actually an important carbon source for TCA cycle metabolism in a lot of different tissues. So then what happened to oxygen levels in these tissues? Well, we worked with Pedro Cabrales in the bioengineering department, and we looked at oxygen levels in a range of different adipose depots, and we found that these were decreased. And this has been shown by a number of other labs before as well. And we also found that blood flow to the adipose depots, or to most of the adipose depots, was also decreased by a high-fat diet. So this, combined with the tracing data on the previous slides, indicates that decreased oxygen tension may contribute to the decrease in branched amino acid catabolism in adipose tissue, but clearly there's a lot of other factors also driving altered catabolism in other tissues as well. So in order to further kind of link this alteration in oxygen levels with branched amino acid metabolism and altered branched amino acid metabolism, we decided to apply an intermittent hypoxic stress to our high-fat mouse model. So basically what we did is we took mice that had been fed a 60% high-fat diet for 7 weeks, so that they were quite obese. And then what we did is we exposed these mice to either 8 hours a day of intermittent hypoxia or a controlled version, which is 8 hours a day of intermittent normoxia. So for the intermittent hypoxia mice, they were in a chamber and they were exposed to 5 minutes first of air with 21% oxygen, and then 5 minutes with 10% oxygen. And basically this is a really common mouse model of obstructive sleep apnea, which has a really high prevalence in obese patients. So although this isn't the same as more kind of chronic hypoxia, we thought it was maybe more relevant to the varying oxygen levels that is likely observed in the adipose tissue with obesity. So what we found was that in the adipose tissue of these mice, that oxygen levels failed to recover up to control levels even after being back in normal oxygen for a number of hours. We found that the intermittent hypoxia treatment also increased their fasting glucose levels, indicating it worsened their hyperglycemia. And then when we looked at branched amino acid levels, first of all, when we looked in the adipose tissue, we found that there was an increase in the branched keto acid to branched amino acid ratio, indicating that that step at BCKDH was likely being impaired. And then we also found an increase in plasma branched amino acids. So this further linked that intermittent hypoxia treatment worsened BCAA levels and may also impair branched amino acid catabolism in these tissues. And when we looked at actually levels of BCKDH in these mice after intermittent hypoxia, we found that it significantly decreased the levels of the enzyme, indicating that branched amino acid catabolism was impaired. So overall then, our data was showing that adipose tissue can use branched chain amino acids as a really significant source of carbon for the TCA cycle and for fatty acid synthesis. And both hypoxia and intermittent hypoxia have potential to or can suppress branched amino acid catabolism and may play an important role in modulating this in obese patients. And then finally, just recently there in December, a paper came out from Sam Klein's lab showing that in humans, insulin resistance, branched amino acid levels, and branched amino acid catabolic gene expression correlates with adipose tissue oxygen levels. So this just lends further evidence to the idea that oxygen is an important modulator of branched amino acid catabolism. Okay, so although there was a lot of evidence showing that branched amino acids, elevated branched amino acids correlate with insulin resistance, really whether or not it plays a causative role is still unknown or not well established. And in addition, how branched chain amino acid catabolism supports normal adipose function is also not well understood. So we decided we wanted to go and look and see if branched chain amino acid metabolism produces anything unique within the adipose tissue that might support adipose tissue function. So what we found when we differentiated these adipocytes is that in addition to the changes in carbon use, also their fatty acid diversity increased a lot with differentiation. So basically this is a GCMS profile of a pre-adipocyte, and this is that of a differentiated adipocyte. So you can see there's a lot more different types of fatty acids, and specifically we were seeing increases in the odd chain fatty acids C15, C17, and also these monometal branched chain fatty acids. And this was really interesting to us at the time because it was presumed that any of these monometal branched chain fatty acids present in humans came from the diet, and that mammalian cells didn't really make it themselves outside of the skin. So we wanted to see are these being de novo made through the same process as palmitate in the adipocytes, that they were really making it themselves and it didn't come from the media. So to prove this then, we traced cells with glucose, which as I was describing earlier, gives this kind of very distinctive mass increments of 2 if it's being used, if it's using fully labeled acetyl-CoA. So the branched chain fatty acids and also the odd chain fatty acids had a very similar pattern to palmitate, indicating that it was being made in a similar way. In addition, if we inhibited some of the key enzymes in the pathway, such as ACC, the synthesis of these fatty acids was inhibited to the same extent as palmitate. And also if we knocked out fatty acid synthase, it stopped synthesis of these. So considering then that these were being de novo made, of course we hypothesized that the branched part of the chain was coming from branched amino acids. So to figure out where exactly in the pathway they were coming from, we traced with the different branched chain amino acids and looked at the label incorporation into the fatty acid. So what we found was that, say for example, when we traced with valine, we saw this very distinct mass increment of 4, which was coming from isobutyryl-CoA, which is the first product of, or which is the product of BCKDH reaction. Then when we traced with leucine, which makes a different branched chain fatty acid, iso-C17, again, we saw that product of BCKDH. In this case, isovalyl-CoA was being used. And with isoleucine, which makes the branched chain fatty acid anti-iso-C17, again, we saw that product of BCKDH was being used. And to further kind of show that it was this part of the pathway that was being used, we knocked out the next step in the breakdown of these branched CoAs. And we find that when we did that, it increased the respective branched chain fatty acids. So causing a buildup of this short branch CoA increased the levels of its long chain branched fatty acid. And then also, if we just added the short chain fatty acids to the cell, we were able to increase the levels in a dose-dependent manner. We both say, for example, here, isobutyrate and propionate. So, so basically, short-chain fatty acids, which we might obviously get from the gut or other metabolic processes, can also influence the fatty acid diversity in the cell and are readily incorporated into new fatty acid synthesis by fatty acid synthase. So then finally, we wanted to see, well, are these modulated by factors that play a role in adipose tissue dysfunction? So I previously showed that hypoxia decreases branch amino acid catabolism. And in addition to this, hypoxia also decreases normal de novo lipogenesis. So for here, I'm showing palmitate synthesis. And TNF-alpha is actually also a potent suppressor of de novo lipogenesis. So that combined then this disruption in branch amino acid catabolism and also this decrease in de novo lipogenesis means that both TNF-alpha and hypoxia decrease the synthesis of these branched chain fatty acids. So overall then from our cell culture, really what we're seeing is that fatty acid synthase is much more flexible than what we previously appreciated and can readily use these branched CoAs or propanol CoAs that come from the mitochondria to initiate an acyl chain. And then basically these are subsequently elongated by the normal fatty acid kind of extension employed by fatty acid synthase. And therefore you can get a range of de novo made fatty acids, and this is going to change based on both the pathways that are active in the mitochondria and also the short chain fatty acid environment of the cell. So next we wanted to see if these are actually made in vivo or whether or not they were just an artifact of cell culture, especially considering that it was presumed that these primarily came from the diet. And it also to start determining the functional role of these cells. So we wanted to see, well, what tissues and biofluids have highest levels of these, and do they vary with disease? And while we were doing this work, this paper came out showing that levels of these branched chain fatty acids are decreased in obese patients and correlate with insulin sensitivity. So we knew to look in patients that suffered from diseases associated with, say, the metabolic syndrome. So we worked with Rohit Lumba at UCSD, who works with non-alcoholic fatty liver disease patients, and he had given his patients D2O or heavy labeled water previous to look at protein synthesis in fibrosis in these patients. But we decided to use these samples then to see if we could see deuterium enrichment into the branched chain fatty acid, as this would tell us that they were being de novo made. As this heavy labeled water gets incorporated into new fatty acids as they're being made directly from the water itself or from NADPH. So when we looked at the branched chain fatty acid deuterium incorporation, we could see that they were significantly enriched to a similar extent to that of other de novo made fatty acids. So this indicates that de novo synthesis was also playing a role in humans. And then we looked to see if they changed with disease progression in these patients. So what we found was that when in patients that had progressed to the more detrimental version of the disease, NASH, that they were decreased. And then we also saw that they were present in levels in the plasma of around 10 to 30 micromolar. And this is actually quite similar range to some longer chain fatty acids such as C20 and C24. So they were relatively abundant. And we also found that they were they were negatively correlated with increasing fibrosis and also negatively correlated with increasing glycated hemoglobin. So so after seeing this, and we saw that obviously they're changing with disease and they're de novo made in humans, we went back to mice to see where they were most abundant. So we found that they are present in the highest levels in the adipose tissue, as our cell culture work would have indicated, and they were lowest in brain, so nearly non-detectable. So we wanted to see then was it necessary, or was the microbiota doing a lot of this de novo branched chain fatty acid synthesis? So although we could see that they were de novo made in humans, it's still possible that they were coming from the gut microbiota rather from our own tissues. And to look at this, we looked at germ-free mice, and we found that levels of these branched-chain fatty acids are actually elevated in germ-free mice, and using D2O tracing, we saw that de novo synthesis was maintained. So really, although the microbiota likely influences their levels, it's not the main provider of them. And then we wanted to, then what we decided to do was look at how synthesis of these changed, or quantify how synthesis of these change in high-fat diet-fed mice, and quantify kind of like an absolute flux in each of the main lipogenic tissues. So when we looked at total synthesis of these, we saw that synthesis was highest in the brown adipose tissue and lowest in liver. And this synthesis was suppressed by a high-fat diet. And then because they were so high in brown adipose tissue, we decided also to look at how synthesis changes with exposure to cold. And we found that synthesis is actually increased in mice when they're exposed to cold temperatures. And since we published this, a paper has come out showing how branched amino acid catabolism is actually essential for normal brown adipose tissue activation. However, whether or not branched fatty acids play a role in this is something that we're trying to look into further. So then, how do these fatty acid synthesis change in general with a high-fat diet? So when we looked in the adipose tissue, we found that if you give mice a high-fat diet, de novo synthesis of fat in adipose tissues for all fatty acids decreases. Whereas if you look in the liver, the amount of de novo made of branched-chain fatty acids is decreased, but the main products of fatty acid synthesis in the liver, palmitate, is actually increased. So this indicated to us that these were unlikely to be made in the liver, and instead possibly they were being transported from the white adipose tissue to the liver. And this was surprising to us because the liver really contains what we thought were all the components needed to make these. So high fatty acid synthase rate, and also active branched amino acid catabolic pathway. But there was one part of the pathway we didn't understand, and that was how these branched CoAs get out of the mitochondria and into the cytosol, where they can be used for de novo synthesis. And we found this paper showing that previously this enzyme CRAT can use these branched CoAs and attach to carnitine so they can exit the mitochondria. And levels of CRAT in the liver are actually really surprisingly low, whereas they're quite high in other tissues such as brown adipose tissue. So we thought that this might be the reason why we're not seeing high synthesis in the liver. And what we found was that if we knocked out CRAT in these adipocytes, that it suppressed or decreased branched-chain fatty acid synthesis, but not the synthesis of other fatty acids. So this really indicated to us that CRAT must be an important part of the pathway and the reason why we're not seeing synthesis in the liver. So then if these are being transported from the adipose tissue to the liver, we wanted to see, well, are these released with lipolysis? So we fasted mice for 24 hours, and you can see here that most branched-chain fatty acids levels go up with fasting, indicating they're being released with lipolysis. And then we also induced lipolysis in adipocytes in culture, and we saw again that there was release of these branched-chain fatty acids. So we're still looking for the function of these, but in conclusion, really we think these branched-chain fatty acids are biomarkers of kind of normal adipose tissue function and DNL. And basically in obesity, where you have factors such as hypoxia and inflammation, these are suppressing branched amino acid catabolism and de novo lipogenesis in the adipose tissue. And this is leading to a decrease in circulating branched-chain fatty acid levels, which has now been shown in a number of patients, or a number of papers where patients with obesity, these branched-chain fatty acids are decreased, and also might contribute to the elevated branched-chain amino acids seen in these patients. And what we think might be happening, or what kind of functional role these might be playing, is they may act as a signal to the liver. And we do have some preliminary data indicating they can activate the transcriptional factor PPAR alpha. And in addition to this, there's also emerging data indicating that brown adipose tissue and white adipose tissue can signal to other tissues via metabolites and lipids. And then finally, there's a lot of papers showing now that increased de novo lipogenesis in adipose tissue, which would increase branched-chain fatty acids, is associated with improved liver function and vice versa. So if you inhibit de novo lipogenesis in the adipose tissue, it has a detrimental impact on liver function. So this is something we're trying to follow up at the moment to see if they might play some kind of signaling role between the adipose tissue and liver. And then finally, I just want to end it on, I suppose, some of the key challenges with studying these branched-chain fatty acids and why they're maybe not more widely reported in the literature. So most LCMS lipidomic platforms lack the resolution to identify branched-chain fatty acids in lipids relative to straight-chain isomers. So generally, most of the time, we're blind to whether or not these branched-chain fatty acids are present. And obviously this limits our ability to determine if they're selectively incorporated into specific lipid species and which would tell us about their location in the cell, or also other signaling functions they might play. And then in addition to that, up until very recently, there was a lack of reasonably priced pure fatty acids for feeding studies. So if we wanted to incorporate this into the diet to feed to mice to see if it has any beneficial physiological effects, it was really challenging to do so. But now a lot of progress has been made on both of these fronts in the last couple of years. So I think hopefully we'll see more publications coming out looking at these branched-chain fatty acids and trying to figure out their physiological role. And then finally, I just want to quickly thank Christian Metallo's lab in UCSD and a number of different people in the lab that also worked with me on this project. And then our collaborators in UCSD, Rohit and Pedro, and we did the germ-free work with Janelle Ayers and the Salk Institute. And we've been looking at their role in brown adipose tissue with Dave Girton at UMass. And then finally, in work I didn't get a chance to show today, we've been looking at these in breast milk with Africa Sullivan here in UCD. And a lot of the early work on hypoxia and branched amino acid metabolism was funded by Ajinomoto, whereas later work was funded by UCSD Momiseed. And thanks very much for your attention. And if you have any questions.
Thanks very much, Martina. That was a fascinating overview of branched-chain amino acid metabolism and branched-chain fatty acid metabolism. So if you have questions, I'd encourage you to put them into the chat function and I'll pose them to Martina. But in just to get us started, I have a few questions, Martina. So my first question was really, you showed very nicely with the tracers how the adipose tissue breaks down branched-chain fatty acids and utilizes the carbons to make fatty acids. What happens if that's done in the background of high fatty acid levels? Where does the carbon go?
Where does the carbon go? Well, actually, so first of all, high fatty acid levels do really suppress de novo lipogenesis. So I think in general, the cell adapts to that to make sure it doesn't make excess. But there is one of the reasons why we think the cell, say, makes branched-chain fatty acids is if you don't have that as a kind of an outlet valve from the mitochondria. So say in a high-fat diet, if the branched amino acid catabolic pathway was active, you could potentially get a buildup of these branched CoAs, which have been shown sometimes to react with other enzymes in the mitochondria and actually impair activity. So it could act as an outlet valve. Actually, if you didn't have that outlet valve, it could be bad for the mitochondria to keep all these branched CoAs within the cell if they're not getting used up through the TCA cycle. I'm not sure if that completely.
I think it'd be very nice to show that that's what happens in that context, because a lot of the branched chain amino acid story, we'll say in the context of obesity and insulin resistance, only really holds true if people are at high risk.
Yes.
So if they have a poor diet, so they have a poor diet, so they have a higher level of fat intake, or there's some other risk factors that are occurring. So it'll be really interesting to see what happens.
Yeah, and I think that's what people think happens in the muscle as well, as in like that you're getting partial breakdown of these branched chain CoAs, and if you're not diverting them off, that it might be having some detrimental impact. So that combination of a high fat diet along with alteration in the pathway is like key to what's happening.
Yeah, it seems to be key for their, I guess, their ability as a biomarker as well. And then the other question that I was kind of interested in was, you know, there's a lot of interest in with circulating levels of branched chain amino acids and their association with development of type 2 diabetes and insulin resistance. And have any of those studies looked concurrently at branched chain amino acids and the branched chain fatty acids? Would be my first question. And the second question is, which is better? Which has more informative, the branched chain amino acids or the branched chain fatty acids? Or do you get more information if you combine them? So it's kind of a three-layer question there.
I think they're all telling you very different things. So first of all, to the first part of your question, a lot of papers haven't looked at these fatty acids. They typically look at branched chain amino acids and only branched amino acids. And that's due to various factors such as limitations with mass spec and things like that. Now, I can say that in some of the cohorts we've looked at, branched chain fatty acids do seem to be a more sensitive marker of insulin resistance than the branched chain amino acid levels. So for example, in a lot of high-fat diet or in diet-induced obese models in mice, branched chain fatty acids are altered, but branched chain amino acids are not. So they are consistently altered in humans, but not always in every aspect. And then finally, I think looking at all three or all branched chain fatty acids and branched chain amino acids tells you the most information, because really, as I was saying, branched chain fatty acids seem like a readout of both branched chain amino acid metabolism and adipose de novo lipogenesis. And they do seem to be very specific to the adipose tissue. So I think the branched chain fatty acids are a nice readout of how well or what your metabolism is doing in your adipose tissue, whereas branched chain amino acids really, I think the levels are probably more driven by muscle, although adipose tissue is contributing. So again, I think they're telling you different things about what's going on physiologically.
Okay, very good. And then the data from the NAFLD patients was very interesting, but then how do you square it with the fact that the liver isn't synthesizing them? So how relevant is it now subsequently that you've done the other work and found that actually the liver isn't important for their synthesis, which I presume you thought when you were doing the NAFLD?
No, actually not particularly. Okay. Yeah. So the NAFLD, I think it's been widely shown that there's adipose dysfunction with liver disease. And like I was saying, there's a lot of mouse models showing that they really have this tight interaction where dysfunction in the adipose tissue can manifest itself in the liver. So I think it's natural that in these patients that although they're obviously classified as having non-alcoholic fatty liver disease, that they're also likely to have some kind of adipose tissue dysfunction occurring, and this might be contributing to the disease manifesting itself in the liver.
So and then if you're thinking down the lines then from an intervention perspective, would really trying to target the adipose tissue could be beneficial?
Yes, yes, hopefully, and there has been studies showing that if they can increase blood flow to the adipose tissue, that increases or that improves insulin resistance. So it does seem that targeting the adipose tissue would be beneficial.
Very good. And then you kind of talked about the gut microbiota in producing the branched chain fatty acids. And there wasn't a huge amount, if I've understood the data correctly. But what about the branched chain amino acids themselves?
So yeah, and this has obviously been a hypothesis, and there has been some data supporting the idea that they might contribute to branched amino acid levels. We didn't find a difference. Now, obviously, a germ-free mouse is a very artificial system, and but we didn't find any differences in branched amino acid levels in the germ-free versus SPF mice. But of course, in humans, where it's much more complex, it could definitely be contributing to the elevated branched amino acid levels that people are finding. But whether or not it's a main driver, I think it's really more a mix of also altered tissue metabolism.
Okay, very good. Okay, I think I don't have any more questions here in front of me, so I think I would like to thank Martina again for an excellent presentation. Thank you very much for taking all the questions and the time to explain everything. And I'd just like to remind our audience that this is a seminar series that's ongoing, and our next speaker is in March, and it's Dr. Leanne Sutherland from the James Hutton Institute in Scotland. And she has a very fascinating title for her talk. So it's Mobilizing the Desk Chair Countryside, ACA, How Play Farming Can Change the World. So tune back in March for that interesting talk. So thank you again very much, Martina, and thanks to all the audience for attending.
Yes, thanks everybody.